So, how does penicillin work? Penicillin is well known as a treatment for bacteria, and today we're going to look at how it actually works. And here's a hint. It's by breaking the cell wall. So let's see exactly how it does that. So let's clear our canvas and let's start drawing. So breaking the cell wall of a bacterium is a really good strategy because they have enormous pressure. So let's just remind ourselves, we've got the cell wall like this and the cell membrane inside, and the pressure in a gram positive one is something like 20, it's not very clear, is it? 20 pounds per square, sorry, 20 atmospheres. So that's something, that's a bicycle tyre, so that comes out to, if I got my maths right, that's something well over, that's a couple of hundred psi, pounds per square inch, which is, considering a bicycle tyre rarely gets above 80 pounds per square inch, that's a lot of pressure pushing out. So what's the cell wall made of, and how can we destroy it? So let's have a closer look. So what I'm going to do is I'm going to clear this, and the cell wall is made up of two chemicals called NAG and NAMA. That's why I remember them. And what they stand for is, NAG stands for N-acetylglucosamine, N-acetylglucosamine, and NAMA stands for N-acetylmuramic acid, N-acetylmuramic acid. So probably easy just to remember NAG and NAMA, I think. So the way these are built up is a bit like this. You make up, you join them together like this. You can join them into a string, and so on. Okay, so that's your basic backbone. And then each NAMA has got 4 residues, polypeptide residues, so peptide residues attached to it, like this. And then these also have attached to them, kind of like at right angles, another side chain of 5 peptides like that. And what happens is this residue here can attach to the blue chain from another chain. So let's draw in another one kind of like behind it. So they've got another chain next to it. Each one of these has 3, So 4 of those, and then blue ones going back. And then what happens is these here can join up with the bottom residue of that one. Okay, so let's straighten this out. So that joins up with that one. And so in that way, they all get cross-linked together. So you end up with a blue chain kind of going off in that direction, and then lots of white chains going across like this. So that builds up this lattice. And there are several sites at which these drugs act. I'm just going to tidy this up a minute. And where the action is, so penicillin do you remember I joined this residue here? I joined that one up with the blue one from another one. It stops this joining up from taking place. And so the result is these strands can't connect. So I'm now breaking these strands here and here, and so the wall just falls apart. It's not penicillio, that's penicillin. So penicillin is a member of the beta-lactams. So penicillin is one type of beta-lactam, but there are others as well. And they're called beta-lactams because they have this funny chemical shape. And it's this square ring here that makes it a beta-lactam. So you find other types of penicillins and other drugs a bit like this. The bad news is resistance is not futile, and a lot of bacteria have developed beta-lactamases which actually specifically attack this ring. So just to make sure the story is complete, penicillins are not the only beta-lactams. So let's show you the whole family. Let's just get rid of our structures here for a minute, move them out of the way. To remind you there that the naughty things are doing. So in addition to penicillins, we also have a group of drugs called the carbapenems. This is just for completeness sake, really. The monobactams, and the cephalosporins. So that's how we attack the cell wall.